Okay, so then let's going. I appreciate very much you coming at this time of the day. It's Sunday, 8 o'clock. It's quite a challenge, so thank you very much for showing up. We're waiting for some more people, and they will show up eventually. So let me tell you a little bit about our point of view of how the genome is folded. So what we were talking about here is the problem of how are your chromosomes folded. And what we're going to be focusing on for most of the talk is how the chromosomes are folded in interphase. That is when things are really rolling within the nucleus. And what I'm going to convince you, hopefully, is that we have sort of a view which takes us across species. That is, we want to come up with a model which actually works not only for human chromosomes, but also for prokaryotes. So that is what we are striving at. And on top, we want to explain not just sort of one experiment, but actually we want to see whether our point of view actually works to explain different kind of experiments. And then on top, we also want so those would be sort of experiments which in some sense connect to statistical physics or statistical problems. And but we also want to connect to mechanical problems, because there are also experiments being done on chromosomes exploiting or trying to see what the mechanics is of the chromosomes. So it's something which is a little bit different from what you have been hearing yesterday, but I hope you will be entertained. So this is the game plan. We want to look at human chromosomes in interphase and also in metaphase. But most of the time I will be talking about interphase. We want to talk a little bit about E.coli and yeast, the beast. So, question is then, when we talk about structure, of course, the first question is actually how do you obtain information about structure? If I tell you I want to understand the structure of chromosomes, then how do you measure structure? Well, one way of measuring structure is, and this here is sort of a view of what your DNA or your chromatin looks like. So you have these histone complexes here, you have naked DNA here, and these red sort of dots are supposed to mimic or should portray the image that they're labeled. Labeled parts of your chromosome, and then the point is, how do you obtain information on the structure? Well, luckily I do have some DNA with me, and here's my DNA. And the question is, if the whole thing would be completely random, the kind of experiment you would be doing is you fix a label here, you fix a label here, and then as a function of this genomic distance or the contour length, you would be asking what is the physical distance between these 2 points. Of course, these would be all over the map, and when you average this, then you would find it's 0. Because you just average in distances, and sometimes like in this direction, sometimes that direction, so it must be 0. So what you really would be looking for is you're looking for the mean square distance between these 2 points, right? And then as you sort of change the distance, of course, then the mean square distance would change. And that, of course, reminds you directly of the random walk problem. And as a matter of fact, it is. And then what you should be seeing is just a straight line as a function of this genomic distance or contour length. So this would be one way of looking at this problem. Just label 2 of these points. Now, the next way of doing this is you just label as many as you can. Then sort of it looks like this in high resolution microscopy. And then you can think of this as really being some sort of a liquid. And then you ask, well, if it's a liquid, we know how to measure structure there, because we were looking at the structure factor or the radial distribution function here, which is plotted here on the x-axis, on the y-axis. On the x-axis, essentially now the distance you're looking at. Now, if it would really be a liquid, what you would be seeing is sort of the first nearest neighbor shell, the second nearest neighbor shell, and then it would die out essentially. Then you would see roughly a gas. Now, what you're seeing here is not exactly this. You see, well, there seems to be some kind of structure, and then it simply dies out quite quickly. And this here, the normalization is 1, so this is basically the gas. Now, the reason why you're not seeing these peaks there is because the way this, the high-resolution imagery here works, it's actually a projection into two-dimensional space. Right? It's essentially you have a very high resolution in the XY plane, but then sort of you have in the Z plane, it's not as good. So, and that is projected into the Z plane. So that is why these peaks are washed out essentially, and you only see sort of this decay here. But it tells you that indeed there must be some kind of a structure in there. So that's the second kind of indication that not everything is random inside of the nucleus. Sorry? It's not exactly exponential decay. No, it's not. So the third kind of possibility you have is now, so we started off with just 2 labels, and then we put quite a lot of labels along your chromosome, and now we put also, all the chromosomes basically with stain, we label them, and then what you find is this quite surprising picture here. So this is the nucleus. What you find is, so each chromosome basically has its own color there. What you find is actually they're on mix. Now that's kind of surprising because when you think about this, well, the chromosomes essentially is just a linear DNA, and then you just compactify this by rolling up some of it and shortening the whole business. But then still, it's a linear polymer. Now, if you have a linear polymer, recall from your basic thermodynamics course, now, if I have this polymer here, and if I wiggle here, this monomer here doesn't really know that this one has wiggled. So hence, this means if I have 2 polymers here, then this monomer doesn't know that this monomer here is on a different polymer, and hence you get this entropy of mixing, and they should be all mixed up. Then you could hold the position and say, well, but hold it. I mean, they're not just homopolymers; there are, in some sense, heteropolymers there. But then perhaps there's some kind of an unmixing taking place there. But then on the other hand, the chromosomes are not so much different, sort of chemically. So hence, that argument doesn't really apply. So we need to explain why we see this kind of structure in the nucleus, that the chromosomes are actually in territories. This one is human. This one is human chromosome, or human cell. So now, so we talk now about spatial kind of information, which indicate there's some kind of a structure in there. Now, there's other kind of information that is topological information. So the kind of experiments that people are doing here is the following. So I'm going to have to take out my DNA as well, again. So now the kind of information you find now is you have these 2 points, and as soon as these 2 points sort of touch, what people do is they ligate, they basically make this permanent there, and then they do some fancy footwork, some fancy chemistry here, and then basically they cut this, again, cut the whole thing and sort of unligate it, and then they find exactly which one was which of your chromosome was next to which other part of the chromosome. And what you get then essentially is this one here. This is from the famous Eris Lieberman paper. Now, what you plot then is you plot essentially your one chromosome against each other, and then you get some kind of a heat map showing which parts sort of were nearest neighbors. So where the color is very hot or very, very red, those were very high contacts, high frequency contacts, and so averaging over a huge population, millions of cells. Of course, there's resolution problems, all kinds of problems associated with this, and actually when you unbias the whole thing, picture changes just a little bit. Given all these difficulties, you still find the following fact that there seems to be some kind of domains here, but let's not talk about TADs and all these issues. Let's talk about the fact that you actually see also signals way out off diagonal, which means that you have long-range contacts in there. Okay, so that's the message essentially, which comes from this thing. So we have now 2 kind of informations. We have on the one hand, we have these physical sort of information where we know what the distances are, so we are metric space. And then on the other hand, we have these topological information because nearest neighbor contacts, they can be happening anywhere in space, so hence you have no idea where they happened. So it's just topological information. You need some kind of an embedding, which then maps this back into space. Okay, but the message which we got actually from the topological information is, if you wouldn't have any idea beforehand, and actually we had an idea beforehand how things are organized, the idea which you get from the topological information is that indeed chromosomes are not just sort of these linear objects, but rather there seems to be these long-range contacts and also short-range contacts, so hence there must be some kind of a loop structure in there. So let's try to get this into our picture here. And so what we want to develop now is a model, and so that we start off with our original picture. And of course, we wouldn't be able to model this even on this scale. So what we need to do is, of course, we always need to do some kind of a coarse graining here. So we start off with sort of the original information, and then eventually you coarse grain into this. And then there is some assumption here, namely that we assume that the coarse grain chain still does have extruded volume, which is non-trivial because you've integrated out quite a lot of degrees of freedom, and actually you've washed out some of the extruded volume interaction, and essentially what you're left with really is only a bending energy there, bending rigidity there. But let's for the moment forget about this and just think about this as some kind of a self-avoiding walk. Now, if we have loops in there, then what is going to happen? You see this sort of transitioning from A to Z. So the first thing is when I have a loop like this one here, of course, obviously it shortens my whole chain, right? Now, if I start to loop all over the map and loop and loop and loop and loop, and you see what's happening is the whole thing sort of gets more and more dense, more compact. Actually, sort of it compactifies, it contracts. This is what you see here. You start off with this original chain, and then sort of you put in some interaction which sort of compresses the whole thing. But if you really loop quite well, then you end up with this this blob here, essentially. Okay, so now let's see what our basic assumptions are. The basic assumption is that we have this backbone with a self-avoiding walk, and then we have certain loops in there. And now comes in our take on this. Namely, we say, well, the loops only are there with a certain probability. The reason, first of all, being the kind of information which we have are only sort of on a probabilistic basis. And secondly, these loops do not exist all the time. Rather, these loops are dynamic. They change. And as a matter of fact, you can prove that these loops have to change all the time. Otherwise, you can from if you try to explain certain kind of experiments, it clearly goes wrong. Sure. Sure. Yeah. It's no. It's in there. Yeah, it's all these transcription factories, etc. And I'll talk about later on about proteins that are involved in this. Okay, so now loops actually are ring polymers. And let's talk a little bit about ring polymers, because this basically helps us understand what is happening here. So loops come in different flavors. Of course, they can be isolated rings. They can be catenated like this. Of course, it can be a little bit more complicated. The winding number can be 1, 2, 3, 4, 5, etc. They can be bonded. The bonded one can degenerate into ring linear, and then I can degenerate into a star-like configuration, right? Now let's talk about not all of this, but rather let's talk about these two isolated rings, sort of how they interact. Now the interesting thing is that when you close the loop, that is when now you take this polymer and then close these two ends, things become different. I argued before, if I have this linear polymer, and if I wiggle here, this one, because of this very, very weak correlation, doesn't really know what is happening here. That's, of course, different when these two sort of come together. If I wiggle here now, then all the rest need to know that I wiggled because I have to close the loop. So hence the correlation along this here is much, much higher than in this case here. This is actually the reason why now, if you take two of these ring polymers and try to push them together, they actually do not like it at all. Actually, they are repelled by this entropic repulsion, which you find in there. And indeed, when you look at this in more detail, you see that these two ring polymers, and plotted on the y-axis, there is now the energy. So it's strong repulsion between these. As a matter of fact, when these polymers are usually, when you look at them on average, they're more ellipsoidally shaped. And what they try to do is, so one ellipsoid likes to stick like this, sort of with a long axis here, and the other one with this one. So that they really minimize their contact. They really minimize their contact, different from these linear polymers. Okay, so loops repel each other. Let's look now at prokaryotes. Now, prokaryotes are already loop-like structures, there are rings, right? So like here in E. coli, we have a ring structure. Now let's do the following kind of experiment. We attach a label here at the ORI, we attach a label somewhere here and a label here at TER. And then what we do is we look now at where these labels end up, when we do the following experiment. Now the E. coli is sort of elongated kind of object. And if there would be no order, and if everything would be like sort of polymer physics would tell you, then the labels would be all over the map, and essentially you would be seeing a probability distribution which is all flat for all of the colors. Okay? So now in the textbooks you can find this kind of structure that you have indeed sort of, this one, then when it really comes into existence is really something which looks like this, sort of this innermost ring, and emanating from this innermost ring are these loops. There may have been so plectamines here, but forget about this. It's basically a loopy structure. Now, so far we have been talking about physics. Now let's bring in the biology. Somewhere we have to touch base with biology here. And how we touch base is now the following. Now suppose E. coli, that is the nucleus, would simply be some kind of a chemical reactor. So you would simply chop off everything, just take all of the molecules in there, chop off your DNA and everything, and then stir it well, and then everything should work fine because you have all the right ingredients in there. But obviously that doesn't work. So why doesn't this work? The reason being, if you have this well-stirred chemical reactor, you have all kinds of unnecessary and unwanted kind of interactions in there. In order to disentangle this, and you have lots of them, plenty of interactions in there, in order to disentangle the whole problem, what you need to do is you somehow need to distribute stuff in space so that things can go on here, things can go on over there, and the two don't know really of each other, that this one is going on here and this one is going on there. And now the take is that actually the organization is being done exactly by your chromosomes. This is the reasons the chromosome exists. The reason the chromosomes exist is because they disentangle your chemical reactor in there. So now the point is, if you have a transcription factor, and one transcription factor sort of regulates something over here, what we say is there needs to be some kind of a physical proximity in order to regulate something, right? If it's just, if I have my transcription factor over here and stuff over there, if it would be all over the map, so you have all kinds of diffusion gradients which would have crossed each other, and that would give you these unwanted effects. So we need to have this physical proximity. Now with this ansatz, so with the loops and with the physical proximity, and now ordering your transcription factors such that the ones which regulate most of the others we put in the center, and then we sort of we do a progression to the outside, and less transcription is regulated on the outside there by these transcription factors. So we have a hierarchical structure of this transcription factor network. This actually does the following. It does exactly the trick which you have found in the text, which you find in the textbook. Namely, you have this structure here, this innermost ring. You have this innermost ring, and emanating from this innermost ring are exactly the kind of loopy structure which you find. So that's kind of interesting that this simple assumption of this physical proximity of the transcription factors with the fact that you have already sort of a loop, then loops the whole thing into loops. And now the point is, we know that the rings repel each other. We have these loops emanating from this innermost ring. And since these loops repel each other, what this means is that indeed we have a bending rigidity in there. We have a bending rigidity on the one hand in this direction, and on the other hand in that direction. So now we have this donut essentially, and this donut is sitting inside of a box. And we're very nasty. What we're going to do is the box is just a little bit too small. So what is going to happen if this box is just a little bit too small? Well, the donut is going to buckle. It's going to buckle like this. Okay? So it looks like this. It's buckling. Now we're going to be even more mean, and we're going to press just a little bit more. What's going to happen if this thing is just rigid enough? It's going to flip over like this. And you have these two bonded rings in there. Of course, the bonded rings, they repel each other, and the story goes on and on and on. You get more and more of this happening, and you end up with a braid-like structure. So it has already this elongated structure, which you find in E. coli. Now let's put back the labels, because our original question was, where do the labels end up? Well, as a matter of fact, the interesting thing is that labels actually end up in these different rings here. And now you already can sort of guess the answer. As a matter of fact, hold on a second. They end up indeed at different spots, which you see here. And this is the original experiment done by Paul Wiggins. So you see here the red one, the blue one, and the yellow one, as a matter of fact, is not sort of equally distributed along the x-axis. So here is your E. coli, and he always projects then the position onto the x-axis. So lots of these E. coli are all projected on the x-axis. So hence, they're well positioned, and we have now an explanation why they are, because they end up in these different rings. Yes? It's chemical potential. Essentially, what you have is different kind of chemical potential in space, and yeah. Okay, so much for E. coli, and in the interest of time, I skip sort of that they also phase separate. The question is then, okay, so you have E. coli, of course, is replicating. Sort of, you have 2 chromosomes in there, and then how do they actually then separate into different compartments? Well, of course, entropic repulsion. Now, so the assumption actually works. Now let's see. Well, yeast I skip. As I mentioned, yeast is a beast, but it works here as well in yeast. You see here, this is the experiment showing certain kind of territories in there, and this is the simulation based on our model, and essentially you get the same kind of right kind of picture in here. So let's focus a little bit more on human chromosomes, because that's quite interesting. Now I touch base with the experiments. I was promising you to look at different kind of experiments, and I'm going to tell you about how we can actually explain these experiments. So let's look at the key experiment here. The key experiment, as I mentioned earlier, is you study now this physical distance squared as a function of this genomic or this contour distance. So the results you see here for chromosome 1, chromosome 11 on the short scale, that is on the just a couple of megabase pair scale. And what these guys have been doing here, this is essentially a Wolf and Rills group at Amsterdam. They have labeled so parts where you have high transcription. So this is here the transcription on the y-axis, and your genomic position on the x-axis. So you find essentially regions where you have very high expression levels, and you have regions where there's very, very low expression levels. So now he puts the labels in the high expression and in the low expression region, and he measures this physical distance. And you see here, this is the low transcription region, and you see it's much more compact than the one which is in the highly expressed region. So in the highly expressed region, they're much more loose than in the less, which corresponds a little bit to heterochromatin and euchromatin. Now, the interesting thing is, and here you can make the argument that if your chromosomes would be all random in there, sort of with just a random organization, what you should find is a straight line. Well, here you could argue, okay, so there is something like a straight line here. Here it's already a little bit more difficult to argue that this is a straight line, but it becomes even more difficult to argue that this is a straight line if you look at chromosome 11. Now we look at the long distances here. You see that it bends over and then basically stays flat. But that it stays flat, we already saw, because what is happening is this whole thing here is starting to compactify greatly because of the looping. Right? So when it loops and loops, and at some point, just it cannot contract any longer, and it's just sort of a fairly compact, in quote, a fairly compact globule which you have sit in there. Yes. It comes, yeah, of course. If there would be no self-avoidance, it would simply contract to just a point, and because of the entropic repulsion which you have in there, you just have some kind of an equilibrium sitting in there. Okay, because it doesn't compact completely like for example, if you just have if, for example, you would have an attractive force which would try to avoid which avoids water, for example, so that just everything is squeezed out and just have a globule. Okay, so now you see the effect, of course, which this looping has. It shortens the distance, and this is some kind of a regulatory mechanism which you have. As soon as you create a loop, you bring other stuff together, and thereby, getting things together, which needs to come together in order to come up with your results with respect to the production you want. So here's now, and I promised you to quantitatively compare with the data. So here is the data again, which I was showing you here for chromosome 11, or the ridges, so where you have an increased expression level and where you have less expression. So this is our calculation here for the two kind of regions, and it fits quite well on the short scale, and it does fit well also on the scale of several tenths of megabase pairs. So this picture actually seems to work. The loops actually do explain what is going on. Yes, please. Do I see increase of expression levels? The one before? Well, this is biology. This is which we cannot explain in there, right? This is how evolution essentially organized the whole thing. It seemed to work, and then this is beyond our sort of explanation. Okay, so now let's talk about, and people have been looking at exponents. Let's look at the contact probability here, or the frequency with which you see these kind of interactions, and plot this as a function of the distance of these sort of encounters which you have in there. And when you look at different chromosomes, you get different kind of exponents here, but all is only happening in 1 decade essentially. So it's quite daring really to talk about any kind of fractality there if you only have one decade of a territory to work with. Now, let's see how this organizes actually your chromosome. So I told you that we see these sort of globules, which is in there. And I told you also that we have this entropic repulsion. And the message which was in this one experiment there was that the ones where you see increased transcriptional activity, those are much less compact than the ones where you see no transcription going on. Now, this means that actually you have sort of, some parts of your chromosome which are highly active, and there's lots of loops and long loops in there, and then there are others where you have shorter loops, which are just a little bit more compact. And because of this difference in compactness, actually, and differences in this entropic level there, in the entropy, what they do is also you have an internal separation into little blobs in there. And the experiment to this is shown here, where again, in Rolf van Drill's group, they've labeled different kind of parts of all kinds of chromosomes, and here are shown the results again for chromosome 11, human chromosome 11, the green region, the red region, and the blue region. And this is what you see in experiment here, that indeed they're not entangled, they're fairly well separated in there, and this is our calculation for this. So the model works quite well also for these kind of experiments. So it tells you that the chromosomes internally are segregated into different kinds of regions, which is consistent with what we were telling before, and namely you want to disentangle what is happening. You want to disentangle this into regions where basically you have one kind of chemical potential and things are happening there, and then you have other kinds of parts which you find a different kind of chemical potential. Okay, so we understand there's an interface, but what about in metaphase? And what about the global picture? Well, the global picture I started off with is where you see that all the chromosomes are not entangled, but rather they're disentangled. They are in these chromosomal domains, organized in these chromosomal domains. And now with this picture of the loops, we understand why this is the case. Namely, of course, also on the global space, you have these blobs, essentially. It's like tissue balls, and it's very, very hard to ram tissue balls into each other. Of course, there will always be some kind of an overlap in there, and the overlap seems to be very, very important and very, very almost very deterministically, because you have these translocations where you see in cancer some part translocates from one chromosome into another one, producing cancer, and that you can, it's very reproducible. So there's almost a deterministic kind of organization in there. So we understand now that chromosomes because since they're looped, they repel each other into these different kinds of domains, and we have this repulsive force. And yes, there are loops on all scales in there. Yes. Yeah, the data from the experiments on these chromosome conformation capture technology, those exactly produce this result. Yes. I see. Yes. I see is then chromosome, just a genome-wide kind of information. Yes. Yes, exactly. So that was shown in this graph where you see the same kind of exponential behavior there, right? Okay. So of course, when you do this also in the simulation, this can be seen, namely. Yes. Thank you for asking. What goes into this? With the E. coli, I told you what actually went into the modeling was the biology, and the biology came from the transcriptional network. Now, what here was the input, which makes the whole thing tick, which actually then touches base with your experiment, was exactly the transcriptional activity. So that essentially what we input in the model is sort of the inverse probability for if you have a high transcriptional level, only have just a few loops. And how do you get a few loops? Well, you make the probability for them, if once they come into contact with each other, very low to stick, right? And if you have a very low transcriptional probability, a low transcription taking place, then you make the probability for them to stick very, very high, and that compactifies them substantially, right? So it's essentially what we take is the gene expression level, so this expression level there, and use that exactly in the calculation, just using the inverse, so that the probability, if you see high transcription, then it's low probability for them to stick together. Yes, exactly. Yes. So this is where sort of at some point you have to bring biology in there, and this is exactly where you bring in the biology. Otherwise, everything was polymer physics, but the biology then comes into when you actually use this proximity idea with the transcription regulation network, and here with the gene expression. The bending, this is an interesting point which you raised there. Indeed, sort of bending is another way of regulating your transcription in there. Because I mean, if you just attaching proteins to your DNA, of course, changes the bending rigidity in there, and hence this is one way of exactly, regulating which parts sort of can come together. If you make it very, very stiff, then you say, well, no way that you're going to make it to come together and work together, right? So this is a mechanism, a physical mechanism to regulate this. It just comes later on. Here at this point, at this junction, it was only the transcription level which entered into the calculation. That was enough to explain the results, explain the experimental results, the topological results, as well as these physical measurements. So I can go on a little bit deeper into this, that there are also other kind of experiments which are explained exactly by this. So we don't need any further parameters on this. Just the only thing which enters in there is this coarse-grained model, excluded volume, and then sort of the looping which takes place with a certain kind of probability. And it's dynamic. The looping is dynamic. There's no static loops in there. Maybe there may be some housekeeping things which always are there, but overall, the loops are very dynamic and change all the time. Yeah? Yeah, the calculations are then Monte Carlo, okay, but you could have done this as well with molecular dynamics or so, I don't care. Yes? Yes. No, that's sort of on a level which we are way beyond. I mean, we are on a much higher level, sort of on a coarse grain level. It is indeed important for the sort of the individual loops on a very short scale. Indeed, you're absolutely right. Of course, the nucleosomes are not equally distributed along your chromosome, and also you find they're very highly dynamic, so they come off very easily, and that's also some kind of regulatory mechanism which you find in there, which regulates again sort of the bending rigidity as well. Yeah. Yeah. No, we're exactly looking into this, but that will be looking at a much smaller scale. So here we are on the scale of, you see, almost megabase, and nucleosomes are not really influential on the megabase. Of course, in total, yes, but we are beyond this scale. We are now looking at this smaller scale on this more hundreds of kilobase pairs scale. And then, of course, then the bending rigidity is highly influenced by the fact that you do have nucleosomes there, dynamics of the nucleosomes, etc. Yes. Yes, of course. Yeah. No, absolutely. But once again, I mean, we are beyond this scale, so we only see this on an average. Okay, so let's look. So I was promising you that I was not only looking at this in terms of the interface, but also in terms of mechanical problems, sort of to touch base with other kinds of experiments. And let's now see how the whole thing now translates into metaphase. So here, being a good German, this for me looks like a sausage, and essentially is asking how wobbly, how stiff the sausage is. Right? This depends, of course, on a couple of parameters. It depends on sort of if you bend it. How can you bend this there? It depends, of course, on the thickness of your sausage. Obviously, sort of a very thin sausage, you have very high difficulty of controlling sort of the bending rigidity. If you make it just a little bit more thicker, then of course it starts to have a much higher bending rigidity. And the kind of experiments that people are doing is now they're pulling at both ends and see what the mechanics is of this object here. So these experiments have been done in John Marco's group, and the result is, of course, not too surprising in some sense. So here is the force, and here now is the relative extension. And you see that initially you have this rise, which you see if you have just a harmonic part. So it's just like a spring which you pull apart. And then as you pull further, then of course, all kinds of things happen, namely then certain kind of bonds essentially break. And this is sort of the rupture. And you see this plateau here where you see there's some yield in there. And then eventually again, you come into a regime which is more harmonic. And indeed, the kind of modeling which we have says, well, you have all these loops. Only now what we have not is these long-range loops. So essentially we have cut now the loops at a certain size. What this does is, from this initial sort of globular structure there, so if you develop something which is more ellipsoidally shaped because you lack some of the long-range interactions, these long-range loops. And then when you lose more and more of these loops on this long range, this initially round or spherical kind of structure developed into an ellipsoid, and then this ellipsoid sort of elongates even further, and you enter into this stage. So The loop picture also explains how you can easily transition from a metaphase into an interphase kind of structure, just by losing some of your long range interaction. There is no need really to pull on it. It sort of comes natural by just losing stuff. And as a matter of fact, I mean, most of it must be chemically driven, and this is losing things on a certain scale. And then sort of it just falls apart. So we can explain, and this is depicted here in some sense. So you can also get the mechanics using this loop picture. Yes. No, confinement is on top. I mean, the interesting thing is that I was arguing that, and I was cheating just a little bit there. I was arguing that, of course, if you have these just two linear polymers, they would disentangle, they would completely mix. Actually, that's not true. That's true in infinite space. In confined space, even linear polymers would disentangle and would phase separate. No, because you have all kinds of proteins. I will be talking about the proteins like CTCF, cohesion, etc., which do indeed bind and do the trick of looping in there. No, electrostatics. I mean, way beyond electrostatics there. I mean, the screening length is such that electrostatics don't play any role on this length scale. The origin of the original loops is sort of the dynamic coming together, and the coming together is on the one hand the changing of the landscape. You have to think about the landscape as really not being sort of a homogeneous chemical potential having there, but rather you have patches of different kinds of chemical potentials in there, right? And these difficult chemical potentials then drive sort of the change in there. And on the top, you also have, of course, all the thermal fluctuations, which are in there, which then sort of bring stuff together. So on the one hand is the forces due to the chemical potential. On the other hand is the forces coming from essentially the entropic and thermal fluctuations which you have. So the density fluctuations which are in there. Okay, so here again is sort of results on these mechanics, and indeed it works quite well. So now you were asking essentially what are the roles, and you were asking what are the roles of proteins in there. Well, the proteins are exactly the ones which sort of produce this kind of binding in there. And there are a couple of candidates which are supposed to be the master weavers of the genome, as they're called. And the master weaver of the genome is CTCF, the zinc finger protein. It's essentially the protein looks like this. Protein sort of looks like this. And the true story is it's essentially an unstructured or intrinsically disordered protein. The CTCF, which is quite interesting because usually you think that only the structured proteins do the real thing. No, here it's an intrinsically disordered protein which does the trick. And it's the zinc ion, which is sitting here, which sort of coordinates these zinc fingers there. And the zinc fingers are linked by very, very flexible linkers. And it looks like this. So here is the zinc ion with coordinating the structure of the zinc fingers, and in between you have these linkers, and at the end of the day it really looks like this sort of. Here are the zinc fingers, and they are linked by these linkers here. So it's really a linear polymer which you have there, which is responsible for, as in many papers they're called the master weaver of the genome. So we need to understand a little bit more about this, and the interesting thing is when you look at how the zinc finger protein actually is interacting with DNA, then you find some very, very interesting things. The first one is now it slides along the DNA, which is not too surprising, sort of the linear polymer, and it can slide along the DNA. And what you find is you have sort of these loops here, and then you have some dangling ends. You will be seeing this much better here. Let's see. At some point, I lost my pointer. So the video doesn't work, but let's see. So you have these tails here, sort of, they're not really bound to the DNA. So this is the DNA, and this one is here, the zinc finger protein. So you have these tails. So it's not that the protein is really bound completely to your DNA. You have these tails. Then sort of you have what we call trains, where here you can see this much better, a train where it really is flat bound to the DNA, and you have these loops in here where it's not bound to the DNA. So it's not like you have a huge sort of binding affinity that really sort of pops it onto the DNA. Rather, it's fairly loose, and it can develop these loops, trains, and dangling ends right there. And of course, from a theoretical point of view, that's pretty understandable, because if you have these, and suppose this one here, this rod here is supposed to mimic your DNA, because on this scale the DNA is much more stiff than this protein. You can think of this just as this rod-like structure. Then, of course, in order to really bind a linear polymer to another linear polymer, which is just a rod, is extremely difficult. You have to have a huge binding affinity in order to counter all the entropic effects that take place, right? And you don't really want this to bind that strongly to the DNA, because you wouldn't be able to rip it off again, right? So you need to have a binding affinity which just makes it tick, which makes it attached to the DNA. And then that's it. And of course, you always then get these loops here, and you always get these trains, and you always get these dangling ends here, these tails. Yes. Yeah, on the short scale, yes. And the electrostatics and the electronic structure is even more important. So, and this is going to be my last point, because we now have an ab initio model, sort of just looking at the electronic structure to predict where CTCF, once again, CTCF is supposed to be the master weaver of the genome, where CTCF is binding. So we don't need any machine learning to understand where CTCF binds to DNA. As a matter of fact, CTCF binds to lots of places at DNA. We understand this now from an electronic structure point of view, from really sort of first principles, where CTCF binds to DNA. So we start to develop an overall picture of what is going on, sort of from the point of view of the proteins, which make the whole thing tick, up to the overall scale, what is happening on the overall scale. Yes? Sorry? Because we want to predict exactly where CTCF is binding. So, and for this, we need this quantum mechanical models, look at the electronic structure and see, are there any signatures in our sequence, in the DNA sequence, which sort of point to the fact that CTCF is binding. Yeah, but here we just look at the signatures, the electronic signature for binding here. Not the electrostatics then. Well, there's certain homos and lumos, so the with the eigenlevels of the. Yeah. So, now, just to wrap up, and I think I'm in time, what we do is we basically bring together physics and biology by inputting into our physical model the necessary biology, or what we think the necessary biology is. And then we can explain sort of a whole range of different experiments. In human chromosomes, we can explain these fluorescent in situ hybridization kind of experiments. We can understand these biochemical experiments with 4C, high C, etc. also the partial genome staining experiments, the whole genome staining experiments. So the partial ones was sort of the unentangling internally, and the whole genome is the essentially sort of disentangling of entire chromosomes. We can explain the mechanical data, and we have also shown that for E. coli, the same model holds as well as for yeast. So we have, I think, sort of at least an idea of what is happening there, what are the guiding principles behind the organization of your genome. And with this, I'd like to thank you for your attention.
I think we have time for a few questions. We ask a lot, but please. Yes, please.
Continuously, do you reproduce the high C sort of contact matrices? Instead of chopping it up into 3 blobs, you have, if you do it at a much finer level, because I'm assuming that it looks at the transcription level data is at a much finer level, then can you actually reproduce
I think nobody really has yet reproduced exactly the high C and the high C maps with a correct model. Because the question, there are multiple questions in there. First of all, is the high C mapping is something which averages over a huge number of different cells. So this is one problem. The second problem is the resolution which you are looking at. There is another problem there. So I think you can only sort of compare overall and gross features. We're not.
For example, TADs and so on would not.
No, we're not. Well, TADs perhaps, but nobody really has a really good model. Neither us nor any other one, to my opinion, to really explain.
Okay. More questions?
They're fairly long, because otherwise, I mean, of course, as always, when you deal with polymers, there is the question of relaxation times, right? Do you have really sort of looked at it long enough so that you have enough conformational changes to make this whole thing, which is even more a problem now, because you constrain the polymer to some extent, right, with these loops, and then of course the relaxation times are really very, very long.
Yeah, but then, I mean, you have to introduce...
Which means that the chains are fairly short.
Yes, but you have to introduce those contacts that you introduce by the transcription regulatory network. So I'm wondering how localized these contacts are.
No, I mean, we take pains, of course, to see whether we have enough conformational changes and enough sort of rearrangements of the furniture with respect to the loops. We of course look.
Maybe if I can I ask another one?
Sure.
I mean, since the approach to including the biological constraints or the biological information in E. coli and in the human case is different, have you tried the other case in E. coli that you say, well, we take transcriptional activity and assume that the more active ones have less contacts? I mean, there are claims like this, that they are on loops that are closer to the membrane.
Yeah, what I haven't talked about is indeed, I mean, you were mentioning now the membrane. What is very important, another regulatory factor, of course, is how the interaction with the membrane is, because there are quite a lot of experiments now showing that there is a lot of interaction with the membrane. As a matter of fact, we have another model where we see sort of where we show that this model is also able to explain the turning which you find in these receptor cells, because there you have an inverted structure in the nucleus. And this is because of the interaction with the membrane. There's sort of a percolation kind of transition where if you have enough of these interactions with the membrane, then sort of just flips over into this inverted structure.
Okay. So let's have the last question.
When you define the monomer length, you said in terms of relaxation time, but isn't it more appropriate to say that this must be of the order of persistence length or Kuhn length?
But nobody knows what the persistence length is. As a matter of fact, recall that Binder and I forgot the person who was working with him showed that persistence length actually doesn't exist. There is no exponential decay in self-avoiding walks. There is an algebraic decay. So that sort of renders the concept of persistence length a little bit shaky.
Okay. Okay, let us thank Professor Heermann again. Okay.